Hello and hello everyone, welcome to class 11 biology. So far in our chapter number 9 biomolecules, we have discussed about two important biomolecules. They are proteins and carbohydrates. Proteins the carbohydrates proteins, they are made up of amino acids. Amino acids are the building blocks of proteins. Right? Protein amino acids amino acids micromolecule proteins macromolecules Then regarding carbohydrates, we discussed about monosaccharides, disaccharides, oligosaccharides and polysaccharides. And among them, monosaccharides, they are the simplest sugar, right? And monosaccharides, they are the building blocks of other carbohydrates. Monosaccharide ni jar wain bana disaccharide chagiwa. Monosaccharide mangbo shoes jar wain bana polysaccharide chagiwa. So we have discussed about those two biomolecules. Now in this particular video, we will discuss about another very important biomolecule, that is nucleic acids. Nucleic acids. There are two types of nucleic acids, DNA and RNA. You must have heard about them, right? DNA and RNA. They are nucleic acids, right? And DNA and RNA, they are made up of nucleotides. DNA and RNA, they are nucleic acids, and nucleic acids, they are made up of small subunits called nucleotides. Nucleotides, they are the building blocks of nucleic acids. Nucleic acid nucleotides ki. So first we will discuss about nucleotides and then we will discuss about nucleic acids. So what are nucleotides? Nucleotides, like I said before, they are building blocks of nucleic acids. Nucleic acids like DNA and RNA, they are made up of nucleotides. Nucleic acid nucleotides. So nucleotides are the building blocks of nucleic acids. But what are nucleotides made up of? Nucleotide a nucleotide, a typical nucleotide is made up of three components. First one is a nitrogenous base. A nucleotide should contain a nitrogenous base. Second one is a pentose sugar. Third is a phosphate group. So a nucleotide is composed of these three components: nitrogenous base, pentose sugar and a phosphate group. Now look at this particular diagram. So this is a nucleotide. It looks complicated, but if we break it down, it will help us understand easier. Now if you look at this green ring like shape, okay, so this is a nitrogenous base, right? And then if you look at this brown, light brown colored ring, this is a pentose sugar. And if you look at this particular blue circle, this is a phosphate group. So a nucleotide is made up of these three components. Now let us discuss about them one by one. So the first component of nucleotides is nitrogenous bases. What are nitrogenous bases? Nitrogenous bases, they are heterocyclic basic carbon compounds containing nitrogen in them. Right. So these nitrogenous bases, they are called as nitrogenous bases because they have got basic property. They are biomolecules. That means they are made up of carbon, hydrogen, oxygen, but along with that, they are also made up of nitrogen atoms. If you look at these two diagrams, you can clearly see over here, along with carbon and hydrogen, right? They have also got nitrogen atoms in them. In your textbook, these two diagrams are given: adenine and uracil. These two are nitrogenous bases. Right. Now, nitrogenous bases in nucleic acids, they are of two types. Those two types are purines and pyrimidines. Nitrogenous bases la purines and pyrimidines. Examples of purines are adenine and guanine. Purines na example ni khare adenine da guanine. Tadige ming dige chema nyayar da. Examples of pyrimidines are thymine, cytosine and uracil. So these three are pyrimidines. Right? So one example of each is given in your textbook. If you look at this one, adenine is a purine. Adenine dikhare purine. Uracil is a pyrimidine. Now if you compare between these two, you can clearly tell the difference between purines and pyrimidines. Purines, the pyrimidines ni para la khewadit tinginyil tana hakur remu tinginyil. If you compare them, you can clearly see that adenine is much larger than uracil because it contains two rings. Adenine, which is a purine, it contains two rings, whereas uracil, which is a pyrimidine, it contains only one ring. So the example, purine la ring ni che, pyrimidine la ring chik. The purine, pyrimidine le chair. Purines are larger than pyrimidine. right? So this is very important. So nitrogenous base, there are two types, purines and pyrimidines. Examples of purines are adenine and guanine. Examples of pyrimidines are thymine, cytosine and uracil. So dige ming dige kena tema nya yarta. The next component of nucleotide is pentose sugar. So what is a pentose sugar? Pentose sugar is a sugar with 5 carbon atoms in it. Remember when we discussed about monosaccharides, I told you that those monosaccharides which are made up of 5 carbon atoms, they are called as pentose sugar. In the nucleotide, there are two types of pentose sugar, ribose and deoxyribose. Ribose and deoxyribose are the two pentose sugar present in nucleotides of RNA and DNA respectively. That means even though RNA and DNA, they are nucleic acids, their nucleotides are slightly different. The nucleotides of RNA contains ribose sugar and the nucleotides of DNA contains deoxyribose sugar. Tadi nenga chamburda. RNA gi nucleotide nanglo la sugar dikharela na ribose re. DNA gi nucleotide nanglo la sugar dikharela na deoxyribose re. Now let us look at these two pictures. Right, this is a ribose sugar and this is a deoxyribose sugar. The difference is quite obvious. If you look at carbon number 2, the carbon numbering this is not arbitrary. So carbon 1, 2, 3, 4 and 5, it is a pentose sugar, right? So carbon number 2 gala sida. On carbon number 2 in ribose there is a OH group. But on carbon number 2 of deoxyribose, that OH group is missing. There is only HO is missing. So therefore this is called as deoxyribose. So this sugar, this pentose sugar is present in the nucleotide of DNA, and this particular sugar is present in the nucleotide of RNA. So pentose sugars. Now at this point in time, you should know that a nitrogenous base attached to a sugar is called nucleoside. Okay, nitrogenous base ta sugar gala taimba nucleotide ki component ni di nitrogenous base ta sugar ta kesi nitrogenous base chik da sugar ni jarwa imba na. A nitrogenous base attached to a sugar, the structure is called as nucleoside. The nucleoside lash the term the so nucleoside is equal to a sugar plus a nitrogenous base. Okay, so we have nucleosides like adenosine, guanosine, thymidine, uridine and cytidine. So these are all nucleosides, right? In your textbook these two diagrams are given. Kesi adenine di ani ribose sugar la jarwa yimba na kharcha dugre adenosine. It is a nucleoside. Kesi uracil nitrogenous base ribose sugar la jarwa yimba na ko kharcha dugre uridine. It's a nucleoside. Nucleoside la term dikhe murda. But kesi dilese rota wa yimba na, here you can see that this adenine and nitrogenous base it is attached to the ribose sugar. So this is adenosine. Same adenine, if it is attached to deoxyribose sugar, then the deoxyadenosine. So this is a nucleoside. Right now it's not nucleotide yet. Which component is missing? The phosphate group is missing over here. Right? We have got only two components, nitrogenous base and sugar. So this is called as nucleoside. It is not nucleotide yet. The third component of nucleotide is phosphate group. Phosphate group, also called as phosphoric acid, is a very reactive group and it is made up of a phosphorus atom in the middle linked to 4 oxygen residues. Right? Look at this particular structure. So phosphorus atom is in the middle and it is linked to 4 oxygen atoms. And some of these oxygen atoms, they are carrying negative charge. Okay, so this is the phosphate group. Now, if you attach phosphate group to the nucleoside, nucleoside phosphate group the nucleoside la domba yimba nucleotide. If a phosphate group is also found esterified to the sugar, they are called as nucleotides. Tadi esterified la disik de la mashia. They'll tenda khala gore la yimba na. Tanjinya la de phosphate the me yimba na nitrogenous base the pentose sugar ni cheku jar de na ko kharimbe nucleoside imba. Ta kisi pentose sugar digala phosphate group ji jarwa yimba na anta shine ko kharcha re nucleotide chare. So nucleotide is equal to nucleosides and phosphate. Nucleosides plus phosphate, it will become nucleotides. Ani nucleotides la ming manda ta nucleoside la thanji ming manda mabuwa. Ta de nucleotide chasana ming change kharcha gare adenilic acid. This is adenilic acid. And then khare thymidilic acid, guanilic acid, uridilic acid, cytidilic acid. So they are all names of nucleotides. Nucleoside laming manda or nucleotides laming manda adwa. So this is a nucleotide. Now esterified latsik de la mashala so di thunda khalla gore law yemba na phosphate group da nitrogenous base ni di sug ju desha kimala pentose sugar di ju de do. If I am the pentose sugar, I am holding nitrogenous base in one hand and phosphate group in another hand. The hand laga chema do khwal jar de carbon la jar de ro. So pentose sugar gi carbon thangbo de la khar ju desha khari nyam do bond form chigido nitrogenous base nyam do bond form chigo. Carbon ngawa jo mare then the carbon 1 2 3 4 and 5 carbon ngawa de la ni khar ju de phosphate group di ju de. Now the bond between fifth carbon and the phosphate group is called as ester bond lower. The imbasonza de la esterified like so the bond between the phosphate group and the fifth carbon of the ribose sugar is called as ester bond, and the first carbon and the nitrogenous base ni para bonds to dilanga jo khari lagire law imena and glycosidic bond. Okay, so these are the two bonds present inside the nucleotide. Okay, so that is all about nucleotides. Let us now discuss about nucleic acids. Nucleic acids are macromolecules. They are polymers of nucleotides and therefore they are also called as polynucleotides. Nucleic acid khari group sha yore nucleotides ke. Nucleotide mangbu chi thu caption khar cha du gore nucleic acid cha du gore. Therefore nucleic acid laming shimba ji khari yore law imba na polynucleotides ke law. The nucleotide dinde do de mangbu shu ji pha su bond form che je an khar cha du gore nucleic acid cha du gore. Yamane polynucleotide cha du gore. So nucleotide ni parala bond khandis form chi do zo de ta right tinenga chumbore. In this particular diagram, in this particular picture, you can see there are three nucleotides. Nucleotide sum do. Nucleotide sum ja khar chi khonzo pha zo thu de do. Ti thu de khari bond khari ge thu de re phosphodiester bond lager. So two nucleotides are joined together by a bond called phosphodiester bond. Now remember, amino acid ni the peptide bond ge thu yo. Monosaccharide ni the glycosidic bond ge thu yo. Nucleotide ni khari ge thu gore phosphodiester bond ge thu gore. The bond dige ming khecham burda. Right. Now there phosphodiester bond khajin la gore na dai la ni ro. Ester bond ni de sida. Nucleotide D gi sugar da nucleotide gang gi sugar ni di parala phosphate group di khachi do link form chi do phosphate link form chi do di link di form che da khande resha carbon da di phosphate di parala ester bond chi che carbon di da phosphate group parala ester bond ni di che so ester bond ni du zambala dil khalla re phosphodiester bond lar ani bond form che da sima do sugar di ga ji gi carbon ngawa da sugar di ga ji carbon sumba ni parala bond form chi gorda. So therefore you can see that there is a formation of a chain of nucleotides over here. So this is a polynucleotide. Okay, so polynucleotide change form chesa vejela there is a polarity. Now remember tinenga chambure polarity dikhare la wimena de la khardu circle din nanglo la 3 prime the dash de la prime le 3 prime de la 3 prime di free reto. So de la 3 prime che the gang de la 5 prime fifth carbon di third carbon fifth carbon. So 3 prime 5 prime. So there is a polarity. Here nucleotide gi change yo bi na nucleotide chain de la khariore polarity yor side chig la khariore 3 prime yor side chig la khariore 5 prime yor. So this is the polynucleotide chain. Okay, so this is phosphodiester bond like this. Like I said before, nucleic acid is of two types, DNA and RNA. Now, what are the differences between DNA and RNA? Let us try to understand that, right? DNA, the RNA, para khewa denga zo ha kwa chenda. First difference, kuni full form de. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. The second difference is the nitrogenous base composition. Inside DNA, we have got thymine, cytosine, adenine, and guanine. In RNA, we have got uracil, cytosine, adenine, and guanine. So instead of thymine, in RNA, we have got uracil. DNA nangla thymine yore, RNA nangla thymine gisabla khare yore uracil yore. So that's the difference in nitrogenous base composition. And then, like I said before, pentose sugar khewa yo. The pentose sugar present in DNA is deoxyribose sugar, and the pentose sugar present in RNA is ribose sugar. Okay, so that is why the name deoxyribonucleic acid like khajin tar lana dinawlal pentose sugar dikhare deoxyribose sugar re RNA la ribonucleic acid khajin lagor lana koi nawlo pentose sugar dikhare ribose sugar. Okay, now DNA is double stranded, usually double stranded. Single stranded yo mare logo martha. DNA is usually double stranded, whereas RNA is usually single stranded. Okay, RNA can also be double stranded, but it is very rare. mostly RNA are single stranded. Single stranded lana khala ore nucleotide chain chigma du stubiomer. DNA dikhare nucleotide chain ni gdupyor. So these are the differences between DNA and RNA. So these are important. Okay, you should remember this. Now, finally, I have one assignment for you, right, which is very important for this year as well as for your next year as well. Right, now assignment is you should read about the structure of DNA. The DNA structure DNA structure. Right, now you have to read in detail about the structure of the DNA and write a short note on the model of DNA proposed by Watson and Crick. DNA structure model scientist Watson and Crick the model the note. Okay, so the tini right. So this is the assignment of the week. Now next we will discuss about another biomolecule, that is lipids. So read about lipids and then you can watch the next video.